There we go.
Perfect. So this is like the only real science part of the presentation, and I'm trying to keep it as basic as possible. And this should be intuitive to your experience as well, having lived with this. So we call SELENON-related myopathy an autosomal recessive disease, meaning that generally you have, with some exceptions, an unaffected father and unaffected mother who are both carriers. And then each parent randomly passes on either the normal copy or the disease-causing copy to their children. So if you take an average base case, out of 4 children, you would predict about 1/4 of the children to inherit both the disease-causing copies, and that child would be affected with SELENON disease. Half of the children, these 2 over here, would inherit 1 normal copy and 1 disease-causing copy. And so they would be unaffected but carriers. And 1 sib would inherit both of the normal copies and would wind up neither being affected nor being a carrier. So that's the sort of very basic math underlying some of the higher level predictions. Any questions about any of that? No. Okay.
Does that work to go on? Or I'll just do it.
Thank you. And what one distinction that it's helpful to make, and that Malcolm made at the end of his talk yesterday in the scientific portion of the conference, was there's something in population genetics, or population level genetics, that we call incidence versus prevalence. And incidence just means how many children at any given time are being born with the disease. Prevalence, on the other hand, is how many people of any age are walking around with the disease. So a sort of crucial assumption underlying the next, the things that we're going to be talking about is that the incidence and the prevalence are not going to diverge very much, because lifespan, we're assuming, and it's a big assumption, is relatively normal, as opposed to some of the more severe end of the congenital muscular dystrophies where lifespan is definitely severely affected. In that case, you would expect the prevalence to diverge significantly from the incidence. Does that make sense?
Yes.
Yeah. So if we assume, and a lot of this requires certain assumptions to even make these calculations, assuming certain population characteristics and certain disease characteristics, we can infer from something called the allele frequency, which is how many disease-causing copies are present in the population, in carriers, etc. We can infer from that how many people we would predict to be walking around with the disease. Okay, so that's what we're trying to do here.
Next slide.
Yeah.
Okay.
So this is sort of the overarching question of this work, is how many people do we think there are with SELENON-related myopathy worldwide? And I want to pause for a second and remind ourselves why this matters. So it's difficult to get an accurate estimate of these kinds of numbers epidemiologically because of a lot of variation in the clinical presentation. I think, Lainey, as you mentioned yesterday, there can even be variation within a family. So imagine how much more variation there might be out there among the entire population of people living with this disease. And we think that there are some milder cases that might be getting missed clinically, as probably speaks to you based on the diagnostic odyssey that many people go through. And then there's differences based on where people live and what kind of access they have to subspecialty care. Is there even a neuromuscular specialist around who has heard of this? And so bringing this back to why we think this matters, the larger and the more accurate of a picture we can get of how many people are affected by this disease, the more of a case we can build for pharmaceutical companies, biotech, venture capital, people who will be able to invest the kind of money and resources that we need to fund clinical trials.
Yeah, sorry. Distracted by the door.
Okay, so again, I've sort of taken out as much of the technical aspects as possible and made this family friendly. So briefly, in terms of methodology, there are some publicly available databases that exist. So ClinVar is one of them, the Human Gene Mutation Database is another, and Nomad is another, which basically are repositories of large numbers of people who have either variants that have been classified, in the case of ClinVar, where you can say they're either disease-causing or likely disease-causing. And Nomad in particular, they've literally sequenced thousands of individuals in the supposedly normal population. And then from that, we can say, oh, how many of those people maybe do not know that they are SELENON carriers? And I'll also point out that there are some disease-specific databases that we're pulling from. So one of them is the CNDIR that submitted about 16 variants that were in common with those public databases. And then the Behzad database, which many of you are familiar with. So we have about 16 variants across the SELENON gene that are in common with these more publicly available databases, and 19 that are private to the database, so not found in these public repositories. And so what we did was we first had to collect all the variants and then run them through a pipeline, and this was done by our friends at the Broad, who cannot be here today. And they run this through a pipeline where they sort of redecide, do we think this is pathogenic? Do we think this is disease-causing? Is it likely disease-causing? Or is it that very frustrating term for so many clinicians and families, variant of uncertain significance? So we have, based on this, a conservative estimate that only takes the ones that we think are likely disease-causing as the basis for calculations. And then we have a more relaxed estimate, which is including those variants of uncertain significance and saying, what do we think is the upper limit of how many people might be walking out there with SELENON?
May I ask a question?
Yeah.
What do you mean by variant?
Exactly, thank you, good question. So variant is just a change in the DNA sequence, which more about them in the coming years, they might get reclassified as disease-causing. So taking that less stringent criterion, we come up with an upper bound of 17,000. So I want to first pause there and ask, the families in particular, how does that number sit with you?
Can you help me with the math of that's 1 in what?
It's just over 1 in a million.
No, so taking the whole population of 7.7 billion, we think that there are whole number, 9,112 people with SELENON disease if these calculations are correct, with all of the assumptions that I talked about.
She doesn't know, as an incident ratio.
Yeah, so I can pull some of those numbers up, but can we advance to some of the last slides? Yeah. I had some backup slides because I thought some people might ask. Oh, okay, maybe we need to go out of presenter view. So I might have hidden them. Let's scroll down.
This one? Oh,
yeah.
This one. I see. Oh, I see.
Those ones, yes. This will be of interest, I think.
Okay.
How about this? Can we right click?
There's no right click.
I'll figure it out. Yep.
And I'll just say to those who are online, if you have questions, please use the meet.ps/Selenon. It's easier for me to manage those questions there rather than in the chat on Zoom.
How about this? Are we able to just project this?
Yes.
Let's try that.
Yeah. Let me just.
And Lainey,
Yep.
can I also suggest, we have only about, we have like 4 or 5 investigators and maybe 6 or family members online. So
Sure.
it might actually just work if we let people speak the way I am now. If it
Yeah.
Sure.
becomes
Yeah.
too distracting or messed up, then maybe we should revert.
Okay.
Yeah.
Sounds
Do
good.
they have a way to raise hands on Zoom?
Yes. Yep. Okay.
That should work. Okay, can people see this?
Yeah, good.
So
Anna,
this is
raise your
breaking
hand.
down, yes.
Anna Ferreiro, you raise your hand. Yeah, you got something?
But well, it was just a comment while you were putting the slides on the screen that to say that these numbers are consistent with what we found and published a couple of years ago, looking at a much smaller population, which is the population in France, where we have quite a particular situation because there is a network of clinical expert centers that cover the whole country. So although we're certainly missing some of the milder cases, we're pretty confident that we're picking up most of the typical cases. And also we have this network of clinical diagnostic, genetic diagnostic labs that work together. So we think we have quite a good coverage of the diagnostics, both clinical and genetics. And we came up with a figure that is a bit above 1 person per million people in the French population. So it's very close to really what you are showing with the more restrictive figures, of course, because these are confirmed disease-causing mutations. So it's very nice what you're doing, very useful to be sure. And also it's nice to see that it's consistent with the French situation.
So to answer your question, the numbers you're looking for are in this column. So that's the global population, predicted conservative prevalence, and this is the breakdown by different countries or different ethnic origins. And we have, these other slides are just the same information in graphic form. So I want to make sure we leave enough time for other people to comment, ask questions, and also for the other investigators.
I'll just say the other point that this particular table here makes, without having to look at any of the numbers, is that based on this type of prediction, we'd expect SELENON myopathy to be roughly similar in its prevalence across different populations. So there's not evidence from here, for example, that Southeast Asians are more likely to have it, or that people from Northern Europe are more likely. So that comports nicely with what Anna was just saying, that we kind of expect this across the world, regardless of somebody's genetic heritage.
There's no evidence to report that any area of like Northern Europe, Southeast Asia, parts of the I worked there. There's no evidence to report that any like area of the world has it more than another. So like France did 1 in a million and they think they have good coverage there. And then this.
Correct. Now that isn't to say that we might not discover at some point, a small localized area where there's been a founder effect, where a single mutation occurred relatively recently, and where there's a cluster of people who are more likely to be affected. Because this is autosomal recessive, that in general, in populations where you have matings between related people, often, for example, in the Middle East, where you may have a village where people tend to have children with their cousins and second cousins, third cousins, there you somewhat tend to see a somewhat higher likelihood. So another one of the assumptions to this analysis is that in technical terms, as a geneticist, we'd say mating is random, but basically the people find their partners without regard to any relationship to each other. And that across the worldwide population, that holds very true. But as I said, it's potential, it's possible that at some point we might find groups of people in one part of the world or another where there's an increased frequency.
And I can add to that actually in early families where I was screening, there was a founder effect in several Turkish and Iranian families, and they were living near the border. So, and there were all the consanguineous marriages. So for sure, in those cases, we will see a higher incidence of the disease.
Consanguineous meaning marriages between people who are closely related.
It's pretty common. And your calculation, Divya, does not take into it assumes no consanguinity, right?
It assumes no consanguinity, and it also, I should add, because it's relying on NOMAD as the sort of base case population for allele frequencies, or how many of these disease-causing copies there are. I should say another big assumption is that NOMAD is not that representative of the global population. There are definitely some subgroups that are more or less represented than others, and we hope that this will be improved with later versions of NOMAD coming out, including one later this year, which should have, we hope, better global representation.
I'm just going to jump in and add an additional comment regarding the conservative, but not still Divya's thunder with some of the things she'll discuss later on in her slide there. The conservative estimate we see as sort of like the lower bound. So, you know, not taking in other things and things like that. And, you know, Divya will talk about some of the challenges later on in her slide deck. But we see the conservative around approximately 1 in a million, or a little bit more than 1 in a million, to be a conservative estimate. So you could read it as, when you see that conservative number of 9,000 something, that there is at least that many people based on this estimate.
That's exactly right. I think we talked about some of the key limitations. Some of them are technical, some of them are because we're starting with databases that are not fully representative, and some of them are because you know, we're making assumptions like random meeting, which may not be true everywhere. I just wanted to jump to the last slide here, which is, what does this mean for everyone in the room? Big picture, we're probably missing some of the milder cases. If you happen to know or find on Facebook or hear about anyone who may be having a milder version of something your family member has, please do send them our way. We have Jill over here, who can answer any questions about enrollment in our studies.
And let me just broaden that. Also, I don't want this purely to be a self-serving Beg's Lab advertisement, but it's really important to raise your hand and get involved with all research. And in particular, I suspect probably most people here already have, but I strongly urge everyone who's willing to enroll in the CMDIR that I'm sure you all know about. It doesn't hurt to be registered in more than one place, but it's really important to have as many people as possible registered. The other aspect is engage with all of the researchers. So, you know, if you're in Europe, reach out to Anna Ferreiro, who's led a number of studies in France, and in other parts of the world. We have colleagues in Australia that we can refer people to. It doesn't hurt to be involved in more than one research study. What is very helpful is if you give us, you know, allow identifiers, allow us to recognize if you're involved with more than one study, so that we don't double count people, so we can get the most accurate estimates and so on. But one of the reasons this is so important is that, you know, a number of us get started in an academic lab, but then to really turn the corner and turn it into a approved treatment often requires the resources of biotechnology, industry, big pharma, and so on. And Anna's experienced this, for example, with her trials of N-acetylcysteine. You really need the greater resources available from industry. The problem, though, is for an ultra rare condition or disease like this, it's very hard sometimes to convince investors or pharma to do that. There's a number that's often bandied about, which is kind of meaningless, but for the people who control money, it's often the first questions out of their mouth, and that is, how many patients are there? And the magic number is often quoted to be 1,000. And so, supposedly, if there's more than 1,000 patients out there that need treatment, then that makes it worthwhile financially for somebody to develop a drug for it. I think it's a little bit BS to say there's a single number. And I've seen successful development of things where there are fewer than 1,000. And I've also seen a number of cases where companies have decided not to pursue something because, despite the fact that there were more than 1,000 people out there. But in general, in order to be so-called clinical trial ready for a particular disease, there needs to be good information about this. And that's in fact why, for example, Chan Zuckerberg sponsored the Rare as One program, which helped fund the work that Divya just told you about. So I urge you to do that. And I see Anna on, maybe Anna, should we talk a little bit about natural history as well and other aspects of clinical trial readiness? It looks like there's about 10 minutes available before we move on to the next topic.
Well, I can. Well, first, I want to endorse really strongly what you have just said, that these figures are really very relevant for therapeutic development. And the other thing really is to have clinical outcomes, so things that we can measure in the absence of treatment, so that when we have a treatment, we can patients score with this in these clinical items with the treatment compared to without. And this usually arises from two types of studies. One is natural history studies, and the other one are more specific biomarker studies. So we're trying to make progress with both. We recently published what we call a retrospective natural history study. That is, we looked back into the follow-up data that were available for 132 patients collected through an international collaboration, mainly in Europe, mainly in France and Germany. And we could identify data about how the disease evolved. And well, one important element is that it also gave us more information about how the disease evolves later in life, so that we are better equipped now to anticipate the complications in late adulthood in the disease. We also have some figures and some outcomes that we quantified. And yeah, so that would be the one on the left, and at the same, that was ours, and at the same time there was another study from Francesco Montoni's group, also from another international series with a slightly smaller but also very representative. And what we found was, because for a good reason really, which is that the disease tends to remain quite stable over time once the respiratory situation has been stabilized and the spinal complications have been stabilized too. So we would need to perform quite a long prospective clinical trial if we want to see evolution of the disease. But this is something that we have already started discussing. It will take a bit of time if we want to plan it well. And the second thing is that we would like to integrate in this prospective study some of the biomarkers that are coming out of our research using patient-derived cell cultures and also the animal models of the disease. And these are partly funded by CureCMD, by the way. So we believe that by combining the clinical outcomes, that is the clinical parameters that we are now able to measure, and the biomarker studies, we can come up with something that might be feasible within the time frame that is typically planned for a prospective natural history study. But in this condition, it's challenging. And I think what we have so far is not bad for retrospective study. I don't know if you have some specific questions about this.
I have one question. So in the retrospective natural history study, what kind of information is relevant? Is it just patient reported or some of the medical history? And if it is that, like how is the standardization done? Because it could be clinician across the world.
I would need someone closer to a microphone to repeat the question for me because there's kind of echo.
Mind walking through that here?
Mind. Thank you. It's straight here.
Yeah, I had a question about this retrospective natural history study. How is the sort of standardization done? Is it mostly patient-reported information from their history, or is it also some of the medical records which are looked at for standardization?
So this was purely based on follow-up clinical records. So we went back to the charts, mostly paper charts still, and we dug out all the figures regarding the respiratory function tests and the force measurement tests over the years for each individual patient.
Got it. Thank you. And I have a follow-up question on the prospective study. Like, what's the time horizon for that? That is sufficient from a regulatory point of view? Because some of these slowly progressive diseases, of course, they would not show as quick a progression.
So you mean the time horizon in terms of duration of the study?
Yeah, for prospective.
Yeah, well, that's a problem. It's typically between 3 and 5 years. 5 years is a very long prospective study already. It's typically around 3 years. The problem is that depending on at what time or what age a patient would enter this study, chances are that we don't see any difference. For instance, what we typically see is that young children show improvement because they are growing up, they are developing muscles, even though they do it more slowly than their peers. While they are growing up, they tend to gain motor function. And so at the end of their growth, this improvement reaches a plateau. And then there's a long period of stability until the age of 30, 35, and then muscle aging starts to be visible to adapt to the underlying condition. And we start to see changes again in the other direction, so towards less good muscle function. So we would need to stratify the patients in different age groups. And I think for older patients, we might see differences already in 3 years. For younger adults, we might not see differences in 3 years. This does not mean that it would be useless, because even if it's very stable, we would have figures to show how the disease behaves in the absence of treatment. So we would expect from a treatment to consider it effective, that in young adults it leads to an improvement, and that in older adults it can stabilize negative progression, but it needs to be stratified according to age.
Thank you.
I'll just say, Naveen, there's a lot of discussion and some controversy around this because for many years, the gold standard in medical research has been you need to do a placebo-controlled double-blind trial where you enroll thousands of patients and half of them get the drug and half of them get a placebo. And then you follow them over time and you wait to see a difference between the groups. And that's obviously not possible for rare diseases, and also not possible for conditions that tend to be go through different stages of progression versus stability across somebody's lifetime. So historical controls are now more often used. So that's exactly what Anna was just talking about, having this natural history information. And then in particular, for example, with gene therapy, the FDA here in the United States and the EMA in Europe have allowed some trials without, obviously not giving a placebo, because it's not really safe or ethical to do that. But the FDA here at least has started to push back and said, well, we would like to have some concurrent controls. And the problem is in a study where maybe there are 3 patients getting the drug and 2 or 3 patients that don't. Well, if you take 6 individuals at random in a condition like this, they may normally have 6 different courses over the next year or 2 years. And so how do you know whether anything you're seeing is just what would naturally happen or not? And this is one of the challenges. The good news is that the regulators, who actually ultimately are gatekeepers to how these things are set up, are aware of this and they make allowances for it. So the bottom line is the more individuals involved and the longer the time period, the better. And so starting to collect information as early as possible and for as long as possible while treatments are being developed is very helpful.
Yeah, and I thank you, Aaron, for explaining that. I think there is of course a safety versus efficacy, trade-off. So I'm assuming that if there's no placebo or control, then they would at least take a look at the safety portion of it, right? Across.
Oh, absolutely, yeah. Anna, do you want to talk about the trial design that you used? I think any minute now, we can probably move on to you and Esther talking about the biomarkers and drug development and so on. But before that, do you want to quickly touch on how you came up, designed your NAC trial?
Well, the NAC trial ended up, it's a good illustration of what Alan was saying at the beginning of the session, how much we need support, financial support from the industry to really come up with a really significant and informative trial. We were cut short with only 6 patients due to funding issues. So the design was, so I think it will be very useful for us to identify something that is very important in this condition, which I think is biomarkers. So our rationale for designing this trial was that we were unlikely to identify within the time frame of a clinical trial any significant evolution of the clinical situation, but we wanted to measure indicators of the oxidative status, of the biochemical status of the patients, because the rationale was that if we could improve that with the treatment, if we could improve the oxidative balance, then we would be protecting, there would be a good chance that we are protecting the cell components from oxidation in the long term, so that we might be reducing the complication of the disease and also reducing the probability of aggravation changes in some indicators using blood samples on some indicators of the biochemical situation of the patients that I think are going to be relevant for planning for the future. Because again, in the long run, we will be able to improve the disease, the severity of the disease and the progression of the disease. So this was the rationale behind the trial. And I think this is going to be an important tool in designing clinical trials in the future. This is what we call biomarkers.
So before we move on, should we give people a chance to ask any questions about anything we've talked about before, online or in the room? Okay, well, Tim, should we move on to the the next topic?
Yes, that sounds good. Let's see. So we're going to talk now. Yes, the next section is about biomarkers and therapeutic targets. And I believe we have a whole slew of speakers for this section, don't we? I don't know, Dr. Anna, if you want to go first, you're first on the list here. So maybe if you want to start things off, or if you know, if one of you feels particularly called to lead things off, we'll go that direction. But I think I have, boy, what do I have? 5 or 6 different speakers here for this section. Dr. Anna, Dr. Ester Zito, Dr. Behzad, you're going to be talking, Dr. Allen, and then I don't know, Casey.
I think for this session, it might be easier to have just Dr. Zito and Ferreiro.
Okay.
Talk about that, and then at 9:30, with the big lab part.
Wonderful.
I will just take over there.
Okay, that sounds wonderful. Let's do that then. So I will let Dr. Anna and Dr. Esther head things off for us. Thank you.
Well, I haven't prepared any specific thing. I think it would be useful if you have questions and you want to voice them. I'd be happy to answer. Globally, it's, well, for the reasons that I have just mentioned, this protein is particular. It's very different from what we see in Duchenne, for instance, in Duchenne myopathy, where the defective protein is a building block of the cell. Is like it's a situation where that can be compared to building up a building with a few of the key building stones missing. Here in selenoprotein related myopathy, we are in a different situation. There's a defective protein that is regulating and modulating other proteins. It's protecting them from getting excessively oxidized. It's protecting them from dysfunctioning in other ways. So we have more leeway for modulating these processes because there are drugs out there. There are not drugs out there that can bring a building block that is missing into a building, but there are some drugs out there that can boost or that can modulate these processes that we are discussing. That's why I think it's particularly important that we identify what we call biomarkers to measure how this is happening. And this is something that my lab is working intensely on. For this, we use patient derived cells, that is skin cells from skin biopsies that we culture in the lab, muscle cells from typically the diagnostic muscle biopsies, but we also use the mouse models. And working on this, we have found some biomarkers that were included in the first pilot trial, but we found also some other biomarkers that we are analyzing now. We are quite excited about something that I didn't present yesterday because, well, for reasons that we can discuss later, but that we may identify in urine, which is the ideal biological sample that we want to work on because it's not invasive at all. Some other biomarkers we have found in blood samples from the mouse model of the disease. And we have found that these two reflect quite well what we see in the patient muscle cells. And these markers we are working on reflect the oxidative situation of the cell. They reflect the endoplasmic reticulum stress, so how much stress these cells are under. But they also reflect what we call the bioenergetics of the cell. That is, how well is the body of the patient producing energy? Because, as you know better than me, one of the main problems in this disease is fatigue. Fatigue of the arm muscles and of the leg muscles upon efforts, but also fatigue of the respiratory muscles, and particularly of the diaphragm. And we know now through studies in the mouse, but also through studies in patients, that this fatigue of the respiratory muscles plays a big role in the respiratory failure in this disease, and that it is reversible in young children, and then it tends to become more permanent with age. So that the energy powerhouses are not working well. So the cells are not able to use a lot of energy. So we're quite convinced that this what we call bioenergetics defect plays a big role in fatigue, and this in turn plays a big role in the muscle weakness and also in the respiratory failure. So this is what we're trying to work on, and this is what we are trying to identify biomarkers that reflect these processes so that we can measure the impact of treatments My lab is interested mainly in drug treatments, and particularly in repurposing drugs that are already in the market, already used for other diseases. So we are now at the stage of confirming these biomarkers using the animal model of the disease and patient samples, and starting to test the impact of drug treatments on these biomarkers. We're dealing with the ethical approval now, because although urine samples are not invasive, we still need, of course, the approval of the ethical committees to be able to collect them and to analyze them in this research setup. So we're hoping that within, I don't know if this was clear or if it's what you needed to hear.
Yes, that's wonderful. Thank you. Thank you. Dr. Ester Zito, did you want to add to that?
You are muted, Esther.
So do you hear me now?
Yeah, we're good.
Okay, fine. I want to just to show you a slide. So clearly we work with Hannah Ferreiro, but our work is restricted to animal models. So we don't have access to patients, but I agree with Anna that we believe that the main point of this disease is at the level of the mitochondria, even though the protein is in another organelle, the endoplasmic reticulum. And I wanted to show you something that we recently get on the animal model. With Anna, we already analyzed muscle biopsies of patients, and we saw something very peculiar on this biopsies, that there is a delocalization of the mitochondria to a band. So normally mitochondria in muscles are coupled to that is called the calcium release unit. And this is very important to allow the mitochondria to generate ATP. So this delocalization can make mitochondria less efficient from a bioenergetic point of view. And I wanted to show you something, if I can, that we recently got. Okay, I don't know if you see my screen.
We do.
Yeah, okay, so this is a muscle biopsy, is an electron microscopy of diaphragm of mice, sapien 1 knockout mice, are coupled with these are calcium release unit, is a sarcoplasmic reticulum. And that we clearly see in the mouse is that here you see that there are areas completely missing mitochondria. And when we count this is kind of striking. We have half of these couples. And from the other way around, mitochondria efficient to make efficient this mitochondria and let them to do that is called oxidative phosphorylation and ATP production. So our goal through different other genetic models is try to understand if we can get again this normal couple that is calcium release unit and mitochondria with some drug. This is just I wanted to show you to say that we start saying that there is a stress of the endoplasmic reticulum in this disease, and clearly it's true. But the main point, the main issue is this phenotype at the level of the mitochondria. Okay, so I want just to add this point. I don't know if you have any question.
Do you have questions about that?
I know people are very interested to hear, what we're doing to develop treatments, and you've both talked about this a little bit. I wonder, Anna or Esther, if you want to talk about how you go about developing a small molecule screen, just in general terms, and what types of things you're finding here that is useful for that.
Well, there was a question, there was a hand raised that I don't see anymore, so maybe that is solved now.
There was just a comment from Ellen and Sophie saying
Okay.
these images are amazing.
They are indeed.
Yes.
So the problem with identifying useful pharmacological therapies, that is treatments with drugs, is that we don't want to test the drugs one by one. Because if it's a very good one, then we need to go from the cells to the mouse, and then from the mouse to the patients, and then do one clinical trial. And if it's not the right one, then we have to start all over again. That takes years. So there is now the technology that allows to screen what we call libraries of drugs, that is collections of drugs by thousands that are already approved by the MDA or by the equivalent European agency, the FDA, the MDA, that are already in the market, some of them. Others are at the stage of clinical trials already, and that can be repurposed to target this disease. But to be able to do these studies that are called high throughput screening studies of drugs, we need to have a very robust readout. That is something that we can measure, that we can quantify in the cells or in the mouse, typically in the cells, and that we know reflects well the main problem of the disease. It's not just a random consequence very much downstream, that is really as close as possible to the cause of the symptoms in the patient as can be. And then we want to have an assay that can measure that and measure the impact of any treatment on this parameter. And this is what we are working on. So we are using now mainly patient cells. We have I can never say this right in English. Alan, help me out. Miniaturized.
Miniaturized.
Like this.
That's very good.
Thank you. So, of course, patient cells are precious material to us. We can amplify them and grow them in culture, but only up to a certain point. So we need an assay that we can do on a very small number of cells, but still to have it very robust. And this, thanks to a postdoc in my lab, Andrew Ho, we are there now. We can measure some of the parameters with only 4,000 cells per well. So this is, it sounds like a lot, but it's a very small number of cells for us. And the other thing that we need to have is something that can be automatized. That is, that can be put in a robot so that the robot can load the different drugs at a highly, with a high throughput. And this is something that we have also made progress with. We have optimized assays that can be done and read, measured by a robot. So the next step would be cooperating with a specialized platform. There are two ways of moving on now. Either we do the screening for the library of cells, we buy the library of drugs, sorry. We buy the library and we do that in my lab, which is an academic lab, or we partner with a dedicated platform that has the know-how and the flow to do this in routine. And the second is more efficient and is what we're trying to move on towards now. We would like to, for the reasons that Alan mentioned in the session before, that we want to make this attractive for the industry so that they bring in the money and are, want to invest on this disease. We're trying to patent some of our findings. so that they are protected, and therefore we can then go and knock on the industry door and say, look what we have, look where we are, and this is an interesting opportunity for you, and it's worthwhile to invest money on this. So these are the next steps for us. We are at the last stages of validating the first 2 assays. We want to have several readouts, but 2 of them are more or less validated now. We want to complete the studies in patients' blood and urine, because again, this is much less invasive than a skin biopsy or a muscle biopsy. And then we are preparing, we recently got a funding to complete the experiments that are missing for a patent registry, so that we are then in a position to move on to this platform high throughput drug screening.
And there's also a question there for you, Dr. Ferreiro. What are the age ranges that the therapies would be administered to?
I think there's no, of course, as soon as we find an effective therapy, there will be no age limitations, particularly for drug therapies. The risk benefit is not the same as we would have with a gene therapy, for instance. So for drug therapies, I don't think there is an age limitation. As soon as we find something effective, it will be given to all of the patients or proposed to all of the patients. The rationale, though, is that if we can bring the cell biochemical status as close to normal as possible, we will have a more protective, if the processes, the intracellular defects that we are looking at are have different steps, are what we call pathways. So there are different parameters that we can measure along the pathway. Some are more upstream of the primary defects, some of them are more downstream. It is, of course, limited. We have to choose, and we have to choose well. But we have found some options that I think are good enough. The main limitation is really the number of cells that we have to use.
Okay. Thank you. Other questions? Yes. Yes, please, come on up.
Hi there, my name is Alex. I'm an affected individual. My question is, how do these types of research get propagated to practitioners all over the world? Like my neurologist, for example, has no idea about what's going on. So how do you folks interact with neurologists all over the world from this very rare disease?
That's a great question. Anna, you just unmuted. Why don't you take off on it?
So I couldn't.
The question is, how does the information and the new advances and the understanding that we're all collectively developing, how does that get disseminated? And Alex, if I understood correctly, your own physicians are not very familiar with SELENON myopathy. And so how do we as a group kind of disseminate this information so that patients everywhere can benefit from it?
I think the typical way of disseminating this is publications. And I think these 2 natural history series that were published almost simultaneously 2 years ago are a useful tool that you can bring with you to any clinic where the physician may not be very familiar with this rare disease. So now you can take these publications with you. They will give him or her enough information that he will not be completely at a loss, and he will know, or she will know, what to expect and how to deal with it. So that's an important thing for clinical care that you have in your hands now. And maybe you won't find easily a physician who has experience on this disease. But if you have a physician who is open to learning, And to listen to this information, that's already a big thing, I think. They can also get in touch with us by email. It's something that happens quite often, that I get questions from physicians from around the world asking for specific questions for a patient. And this is something that we're always very happy to answer. For the more scientific aspects of things, well, it's a bit more delicate. We have to publish them as well. But as I mentioned, nobody, I think none of us is interested in patenting for making money with this. None of us is making money with this, but we want the industry to be attracted to this disease so that therapeutic development is reality. So for that, we need to patent things before we can publish them. And that's why we have been holding on a bit on our data, because once we communicate about them, they cannot be patented anymore. But eventually it will happen. I think eventually will be months and not years. And then what we do is publishing in scientific journals and also communicating the results in congresses in like this, where patients are present, but also in scientific and medical congresses, so that the disease of neuromuscular specialists can be aware of what is going on.
Yeah.
Does that help, Alex?
Sorry, Tim.
No, I was just checking with Alex to see if that was the answer she was looking for. So that's, thank you.
And Alex, and for everybody in the room, I'll just emphasize, I mean, you as empowered patients can control a lot of this. It's unfortunate sometimes that the overall field doesn't do this spontaneously, but as Anna said, don't hesitate to reach out yourself. Don't hesitate to connect your own physicians with us. And then we can then in turn refer them to, or you to other physicians maybe closer to home who have a little more familiarity with this. So reach out and take charge yourself, be an empowered self-advocate. And I think you'll be surprised how receptive a lot of people are when you raise your hands and ask those questions.
Yeah. I would concur with that, just from our own experience with boys. Yeah, it's a little bit of problem solving and solving a puzzle. So, yes. Any other questions here for this segment before we move on to the next? Oh, yes.
Not a question, but more of a comment to what you were saying. As someone who just finished neurology residency training, we didn't learn about SELENON in medical school. We didn't learn about SELENON in residency. Everything I know I learned from the lab and from subspecialists. We are muscular subspecialists at Children. So to even get a neurologist who is going to know about this disease is challenging, but I would hope that they would be receptive to whatever information you bring to them or any questions that you ask them, and to learn from other people who are more specialized and know more about it.
Thank you. Okay, are we ready for the next segment? So we'll move on to, I think on my agenda is called part 4, but I think it's really part 3 here. But we're gonna open it up to the Begz lab to,
Sounds good.
Yeah, and I'll go.
Hi everyone, my name is Behzad Moghadaszedeh. So SELENON is very close to my heart because it was over 20 years ago I found the first mutation in this disease. It was a benchmate with Anna Ferreiro back then. So it might be very rare, but for me it means a lot. Actually, Alan, I think I'm going to start with my own slide from my talk. It might be easier. Let's see if I can find it.
And actually, while you're pulling that up, let me just say, some of the stuff that we're about to show, in particular the work that Pamela Barraza-Flores will show, is new, but it actually ties in very closely to findings that Ester and Anna have made previously. So I'm hoping, Ester and Anna, that you guys will break in and make comments and thoughts and questions and criticisms. And for everybody else in the room, you're going to get to hear that process a little bit of scientists exchanging data and wondering what it means for the first time.
Sounds good. So clinically yesterday.
Share with everybody.
Oh, is it not shared?
No, here, I'll do it.
Oh, okay.
Got
All
it.
right.
No worries.
Thank you.
Yep, there we go.
Can you see the slide, Alan? Okay, perfect. So from a clinical point of view, yesterday we had several talks with other diseases like collagen 6 and LAMA2, and some of those have like a cardiac involvement that was very scary for patients. So the good news for SELENON, don't have to worry about that. So there is no cardiac involvement in here. So from that point of view, you're fine. And this graph here shows the proportion of patients that can walk throughout their lives. So some patients that have a more severe clinical aspects, they lose their ability to walk. But after that, those who are still able to walk at 20, most likely they will be able to walk throughout their lives. So the scoliosis, this graph shows the proportion of patients with the scoliosis. So you see by age 15 or 18, many of them already have a scoliosis. By far, the most important aspect of this disease is the respiratory involvement. So the trick also with the diagnosis is that many patients who do not have a diagnosis do not know that they need this ventilation. And I think the team and Elaine were talking about this yesterday. As soon as we are diagnosed, you are referred to respiratory care, and it was life-changing right away.
Mm-hmm. Absolutely. Yeah.
So that's something that's probably required throughout their lives. But if this is taken care of, most patients have a very normal and fulfilling life. So I'm going to say a few words about the molecular aspects, and all this again is based on Ester Zito's work that really changed our view in this field. So this shows like basically half of the muscle cells. So this is the membrane of the muscle cell here. This organelle inside the cell is called a reticulum. And Ester Zito showed that the SELENON interact with this protein called the SERCA. SERCA is a channel that lets the calcium from inside the cell to this organelle. So this organelle is where the calcium is stored. And when the muscle is activated for contraction, the calcium is released through this R/R channel. And then when the muscle needs to be relaxed again, the calcium is pumped back constantly by SERCA channel in there. The problem is that when the cells, when there's too much oxidation in the cell, SERCA channel is less active. And that can cause some issues with a muscle contraction or relaxation, but that's the hypothesis. The SELENON job is to reduce that from the oxidation form back to reduced form to make it more active. So that's one aspect of the disease, is the calcium homeostasis here. The other thing also that both Anna and Esther were just talking about is the energy aspect in the cell. So the mitochondria are the powerhouse in the cells. They produce a molecule called the ATP that's used for all the energy aspect of the cell. To do that, to produce the energy, mitochondria needs to come close to the reticulum and get some calcium pumped in. And it has been shown by Ester's Zito's lab that SELENON is also present in areas of the reticulum where you have close contact between mitochondria and the reticulum. And in the absence of the SELENON, the mitochondria moves a little more away and is not as active as it should be. So all this just brings us some hypotheses about what we can do.
Actually, Behzad?
Yes.
Do you want to go back one slide? And just because all of us have slightly different views of things, Esther and or Anna, did either of you want to expand or contradict or give your view of what Behzad just said? Anna, you need to unmute. Or
For me, it's perfect.
Oh, thanks.
Exactly.
Okay, good. So we're in agreement.
Very good. Go ahead.
So based just on what I just said, we try to understand how things work. So one aspect is when SELENON is deficient, it can affect the SERCA's activity, which would affect the calcium homeostasis in the cells, how much calcium is in the different places. And that could possibly affect the way the muscle contracts and relaxes. The other aspect is this alteration in calcium in the cell can affect the mitochondria function, which affects the metabolism and energy production. That could also result in muscle weakness. So the idea is, once we know this molecular mechanism to some extent, and this is a really huge breakthrough that came since 2015 when Ester first showed the interaction with circa. So we have different ways of intervening here. So the first way is to simply correct the original deficiency by gene therapy. Just bring a new copy of SELENON gene to correct the original deficiency. So that's all obviously very promising, but it won't solve all problems. For example, a patient that has an advanced scoliosis probably won't see any changes in that aspects. That's why we have to look at multiple ways to do this. So another way also would be to take care of the oxidative stress. That's what Anna has been trying to do with the N-acetylcysteine treatment, to reduce the oxidative stress. And all the drug screening that was mentioned, we could just intervene at different steps in this process to either boost the circuit activity or to boost the mitochondria activity. For all these, we need models. So we don't see my cursor. Okay. So we have like cellular models, zebrafish models, mouse models, and even ex vivo muscles where we excise a muscle from the mouse, and in vivo, ex vivo, we can just measure its activity and its force. So in the Behzad lab, we are working on all these aspects, and something that I'm very excited about in terms of cellular model is working with iPS cells. They are induced pluripotent cells. So David Mack, who was also in the conference here, he developed a very nice technique. From urine of patients, he can isolate epithelial cells and reprogram them to make them muscle cells. And we can grow them in muscle and actually have a tiny muscle that we can actually measure force and other parameters in that. So that's something probably reach out to some of the patients we have to see more carefully what mutation and what age that would be important. Probably reach out to some patients to do that. It's absolutely non-invasive and it's very helpful. So the cellular models, as Anna said, they are very useful for high throughput screening. So you can screen tens of thousands of drugs at the same time. And again, as Anna said, we can even start with the drugs that have already been approved by FDA. So they have already been approved. So we can repurpose them. If you see any benefit in our system that we will be changing a phenotype or rescuing the lack of a SELENON in any way, that is going to be very useful. And then those drugs that we find in cellular models, they can be, so that will be a high throughput screen. Then we can bring them in a medium throughput in zebrafish models, and then in mouse models, and all these things can be confirmed in the ex vivo models as well. And so right now in our lab, we are actively working with the mouse model, and for the zebrafish model, Pamela is here. I think she can take over and show you. Alan has your poster he can display. Do you have any questions so far? Yes?
Why are some muscles affected differently than others? So why isn't the heart affected where it is?
That's a great question, and the answer is we don't really know. I don't know if Anna has any suggestion, because sometimes some of these muscle disease, they are caused by gene and protein that are expressed exclusively in the muscle. SELENON is expressed everywhere, and it's causing a muscle disease. But one aspect maybe is that because it's affecting the metabolism and the mitochondria function, and muscle is a huge consumer of energy, maybe that's why it does that. But between the skeletal muscle and the heart, why it's not affecting the heart, I do not know. Anna, do you have any ideas on that?
Well, yes, basically what you just said. The muscle is both one of the organs that requires most energy to function, because contraction uses up a lot of energy, but it's also, and this is something we're paying more attention now to, a huge metabolic organ. It plays a very big role in using sugar, for instance, to produce energy, and in using fat to produce energy. A lot of that happens in the muscle. So we think this is one of the reasons why muscles are more affected. And also this architecture of the muscle, as Esther showed, the architecture of the muscle is very tightly regulated. And the different organelles inside the cells have to be in a very precise position so that everything works well. And mitochondria have a very tightly regulated positioning in the muscle, which is unlike other organs. As you mentioned, Behzad, it's a question why in the heart there's no problem with contraction, but the contraction in the heart and the way the calcium is coupled to the mitochondria is a bit different to what happens in the other muscles of the body. So we think this is one explanation. The other explanation is that some of the muscles and those that are more affected in this disease, particularly those in the back and the neck and the posture muscles, they depend more on mitochondria to get their energy, whilst other muscles, such as the thigh muscles, for instance, which remain quite strong, use more glucose, use other substrates, other types of cell food to generate their energy. But we're not sure there's nothing else in other organs. As I said, the biomarkers we find are altered also in blood, and they are altered in the skin cells. So there might be something happening in all the tissues in the body, but it only gives real signs and real problems in the muscle because, as you said, it requires a lot of energy and the architecture is very, very tightly regulated.
And we have a question from Elaine. Does the different structure of cardiac and smooth muscle have something to do with different impact of SEPN deficiency?
Probably yes, for this reason that the coupling between the calcium flux and the mitochondria and the generation of energy by the mitochondria is different in smooth muscle, that is involuntary muscle, and in cardiac muscle compared to the limb muscle, the skeletal muscles. But maybe Esther, you want to comment on this?
So I believe that for sure we don't know why a cardiac muscle is not affected, but I agree with Anna that also regarding skeletal muscle, so the more affected muscles are the oxidative muscles. So in other words, muscles which rely on oxidative phosphorylation to generate ATP. And I believe that this is the main point, but yeah, unfortunately, we are not sure. This is a guess.
And I have a question myself for both Anna and Esther. Have you guys looked at the expression of SELENON in smooth muscle? Because I bring it up because there was a question by Susanna saying that some of the patients who have the problem is swallowing, and we're wondering if that could be due to any defect in the smooth muscle. Any ideas there?
That is a problem with, particularly with evolution of the disease, but swallowing is a complex mechanism that involves not only smooth muscle, but largely also striated muscle. So part of it is voluntary, part of it is involuntary, but the problem is in the upper parts. So the esophagus that is inside the thorax is involuntary muscle mainly, but the swallowing problems and the risk of choking during meals comes from muscles that are in part voluntary and that are in the upper part of the digestive system, more in the throat. So it's difficult to tell apart which is the component. I think this would be something very interesting to look at, and maybe this is where you as patients as well can help us, because we don't always think about asking specifically if there are problems with digestion, for instance, or with other we know there are problems with bladder. There's often problems with incontinence, urinary incontinence, but again, it's difficult to tell because there are smooth muscles and striated muscles involved where it comes from. But I think it would be very useful if you report in whatever way is easy for you, through the CURE-CMD registry or through direct contact with us. If you report anything that does not look directly related to your limb muscles, but is bothering you and you think might be part of the disease, because this smooth muscle involvement can be overlooked quite easily, really.
Thank you. I think we're going to move on to Pamela. This is her poster about the zebrafish work she has been doing.
Hi, everybody. I'm Pamela. I am a postdoc in Dr. Alan Bigg's lab. I'm a molecular biologist, so I mainly play with fish a lot and cells and sometimes mice. So my main goal and objective in the lab, I've been working with Alan for 2.5 years roughly, and the whole goal of my work right now is to generate new ways that we can test drugs, whether that's a big library, meaning a lot of drugs at once, maybe automized, that is the end, like that would be ideal. The more drugs we can test, then it's kind of like approaching the issue with brute force, right? We try 10,000 drugs and maybe one will come up and then we can work from that. But also anything in between is very useful. So I was fortunate that when I joined the lab, they already had a lot of models in the zebrafish, and I was very interested to work with the zebrafish. So I want to talk a little bit about that, but just know that my objective is to create these assays that are pretty much like an experiment that we can run over and over to test whatever we need that is reliable, but at the same time describing how the disease works. Because if we know how it works, we can also narrow down our targets. So that is my main goal. Anything that I will try will be with the purpose of creating this type of experiment while elucidating how the disease works. So one of the big advantages of working with zebrafish is that we can look at the embryos, because the fish, when they lay eggs, the eggs are transparent. So it just so happened that when I was looking into these fish and just testing a lot of things in them, just making sure that I wasn't missing a phenotype, which means how they differentiate from a fish that is mutated from one that isn't. So how can we tell that one is sick versus another one? Because another funny thing about SELENON is that when we put the mutation in their animals, the animal seems to be completely fine. So we've been looking into how can we, where to look, pretty much. So that has been my job. And what I found is that and during the embryogenesis, there was a phenotype. I think we can.
Maybe Alan can zoom on that area of the poster.
Well, I just wanted the video to play, but can we see it? Yes, we can. So
I'll zoom. Which panel do you want me to zoom on?
I just wanted the video to play because there's a Twitch video there.
I think, okay.
Yeah, but it's okay. So when yeah, there it is, perfect. Yay! So when this is a zebrafish embryo, and when this is a 20-hour embryo, so it's only been fertilized for 20 hours. And so something unique about zebrafish is that at this time, I don't know if there's other types of fish, or maybe it's not that unique to zebrafish, but this doesn't happen in mammals. They twitch their little tail at that spontaneously, and I can measure how well they move. And I found that in the SELENON fish, they weren't moving as well as the wild type. So I can use this because these are so tiny. We can use this to test a drug, for example, multiple drugs, because they're such tiny eggs, are like maybe 10 millimeters. So they're very small. And so, but because of that, I thought, well, maybe we should also look into the embryo, because something that we know in the field is that SELENON is used during embryogenesis, but to my knowledge, there hasn't been that much exploration during embryogenesis, even though there are papers that show that fetal tissue not from patients, but just fetal tissue, SELENON is expressed highly in the embryo. So maybe there's something going on during the embryogenesis, and especially because during this time point, these are the precursors of the axial muscles, so the spinal muscle. So I don't know if that has something to do with the presentation of SELENON, but it might be relevant to study them. It's not something that I'm doing at the same time. But in this poster, I'm not going to go into all the data, but I brought 3 different assays that we can test to test for drugs, and I'm also expanding onto researching what's going on in the embryo.
I think we have like 7 minutes left. It's open to any question you may have.
Yeah, any questions?
Also, I'll open the screen so you can see the online people.
And also, I would love to hear from Dr. Ferreiro and Dr. Zito if they have any thoughts about what I just presented, because this is, haven't really shown that.
Did you want to talk quickly about your seahorse experiments?
Yeah, sure. And one of the assays that I developed, this isn't in zebrafish, this is in muscle cells. We have this machine called the seahorse. That's just the name of the machine. It doesn't use seahorses or anything like that. But it's just a little machine that just measures fluorescence. It's nothing too extravagant, but we can put cells in that machine in a plate where we have like 96 little wells, and we can put a lot of cells and a lot of N numbers, and we could potentially put 96 drugs in a plate, for example. So it's a good candidate for moving on to finding drugs. But I use this machine to measure the metabolism of my muscle cells. And so we generated different cells that we mutated the SELENON gene. So we effectively created a cell line that it's a model for the disease. And there we found that respiration of the cells was decreased in SELENON mutated cells, and same as glycolysis. I also verified that by taking cells out of the mouse and putting it in this same experiment. And just because sometimes cell lines can create their own mutations and kind of rescue themselves, so we want to try it in different cells with different organisms. So I also did that, just taking cells from the mouse quads. So I've tested this from muscles from the mice to verify that. Yeah, and this is something that we can already start testing drugs, and we can see if I had a drug that maybe, based on what Dr. Zito and Dr. Ferreiro have done, maybe it can rescue the oxidative stress that they were talking about in the model, and maybe we can see like these data points, maybe they can back to wild type. So this is a way that we can test a lot of those new innovations that they be pushing forward.
Hand raised. Dorani, go ahead.
Hi, yes, I want to thank everybody, but question about how long does it take to test these drugs? You keep saying like you can test different thousands of drugs. Like how long does it take to test some series of drugs, and how many approximately are there to test across?
That's a great question. The hardest part is to design the assay that shows a difference first between our SELENON deficient cells and normal cells. That's the hardest part. Once that thing is set up, it's
yeah.
And in zebrafish is different. In cells is different too.
Do you mean how long does it take, like in the assay itself? Like how long does this particular assay take? Or do you mean like how long is it going to take you to find a drug, kind of like, or test 1,000 drugs?
Yeah, it was more like how long does it take to test 1,000 drugs? There was.
Yeah. I mean, I imagine because these assays, I'm the only one that knows how to run them for right now, especially the cell one. It takes me like a full day to run one, maybe, and then in preparation for them, it takes me like a whole week. And I can test 96 at a time, for example, not including controls. So I imagine that that will take me like a few years. But however, this is kind of like the part where if we validated, we are very sure that we want to move forward with these particular assays, which is why I generated more than one, because we want to pick the best one. Once we've committed to one, then it's something that can be, how do you say, scaled up to industry or other people that can possess like other tools, and they can be compatible with robots, and there could be an automation. That's something that can be, we have a core at Harvard Medical School that can work with drug screen, little, like small molecule drug screening. So it depends on how much we can adapt the assay.
It also depends a lot on the type of assay. Some assays can be already done in a large volume, like 384 plates. And the seahorse, for example, is like a smaller scale.
Right.
So we can start with another different type of assay. And once we find some good candidates, then they can be tested in the other assays that are at the lower throughput. I see also Esther wanted to add something here.
Yes, please.
Yeah.
I'd love to hear your thoughts.
First of all, thank you. Beautiful, beautiful talk. I have just a curiosity. So the fact in cellular respiration that you saw in your C2C12 was in myoblast or after differentiation in myotubes?
Yeah, thank you for asking that. So I tested tested thoroughly only after differentiation. And I saw the effects that you saw when I overcrowded the wells, when I had high concentrations of cells, which at lower concentrations, I had a nice curve when the difference in respiration and glycolysis it increased as the number of cells plated increased. So what you saw is like 20,000 cells in a 96 well plate, which is pretty crowded. To me, it indicates that there's potentially some oxidative stress in addition to the differentiation. And that's when I see these effects, because I also, these effects that you saw were normalized to differentiation. So we made sure that we were taking that into account. So I think that's a big part of it.
Thank you.
Does anybody have like a question that can like maybe make things more simple or, sorry, I didn't know if it was.
Naveen?
Yeah.
In I think one of the talk 2 days ago, they also demonstrated zebrafish form. I forgot for which condition. Does zebrafish form take like longer take longer for a drug to be measured versus, let's say,
in a cell line?
Yeah.
It depends. Again, repeat the question. Oh, did you guys hear the question? Does it take longer to run an assay for these purposes in the zebrafish versus the cells? So it depends. For example, the cells take me a lot longer because I do multiple things after I do the assay to normalize it. But I imagine if it was to be automized, then that wouldn't be a problem. So to me, as one human, it's longer in the cells. But with the zebrafish, I just put, for example, there's one of the assays that I showed that I didn't get into, is like I put the fish in a plate, and then the fish will swim, and they're just recorded and followed by a machine, and we just measure how much they swim. So that only takes me putting them in the machine, recording them, and then the machine just spits out the data. And then, I mean, not just, but then I just get the data and analyze it. So it just depends, but I imagine that scaling up a zebrafish assay is a lot more difficult than scaling up a cell assay. Am I wrong?
Just to be clear, these would be considered very low or at best maybe medium throughput assays. You can only do a small number of compounds at a time. What Anna has been developing is much more state-of-the-art, biochemically focused. You can, in cell culture, you can do thousands, tens of thousands. Pharmaceutical companies will test a library of 100,000 compounds. I don't think any of us are there yet, but Anna is getting closer to it. with the approaches she's taking.
Do you want to add something, Anna? Yeah.
Yes, I just wanted to say, because I think we have to be very as quick as we can, but also very realistic in the expectations that we raise. So when we think we can test 10,000 compounds, but for doing this, we need to pull them together. So we test several compounds on the same set of cells, and when we see a positive impact on the situation of the cell, then we have to go again with separate isolated drugs and test again and then confirm and then reconfirm. So one thing is the time that you need for one individual test, but there are all these stages of refining the results that take a lot of time. So it's not one run. And I think we are hopefully getting there, but it's not something that can be done in a few weeks or even in a few months. And then when we find a drug, we still probably need to validate it in the animal model of the disease. So with all its limitations, I think it's very nice to see the impact of the mutation on the zebrafish embryo. That's a fantastic model for validating whatever we are going to find in the high throughput screen. So, I think we're making very good progress, and Ester's findings in the last years have really helped us move on with the feasibility of these screenings. But I think we have to be very realistic in the time frame. High throughput doesn't mean overnight.
Yes, it does not mean, but it is a faster approach than perhaps
yes,
gene delivery or something that's a biological thing that we're bringing into the treatment.
I'd only add to that, that just as Anna is saying, I wouldn't view these different approaches as competing, but as complementary. Any pharmaceutical company is going to want to see validation in multiple models, not just cell-based, but also animals before even venturing into humans.
And I would add also the molecular screening. So we are looking for rescue, for drug rescue, but the potential drugs we find can also help us better understand the pathophysiology, the mechanism, and come up with new ideas too.
If the more we know about how it's working molecularly, then the easier, instead of running 100,000 drugs, and we can select drugs that we already know target those molecules. So
Find a pathway.
yeah, so that's why it's a double, I mean, two stones, one bird kind of situation, because we learn about the biology of SELENON and we generate ways of testing it, which is how we design our experiments. And in addition to what Divya said of things complementing each other, like what we're seeing in the embryos, the model that Dr. Zito and Dr. Ferreiro have put forward is very skeletal muscle focused. So in the embryo, the mechanism can be, like, the proteins are very different. There is just a completely different stage in life. So if we can find a drug that can work in both worlds, and perhaps there'll be better chances of getting an effective treatment.